I want to welcome all of you to this PolarConnect event. This is Janet Warburton, and I work for the Arctic Research Consortium of the United States. I'm based here today in Anchorage, Alaska, and it's not such a pretty day on this August 8th, 2016, a little bit overcast, but I am very excited to be hosting this webinar where we're going to be talking with PolarTREC teacher Sandra Thornton and some Arctic researchers that are currently all sitting, I believe, in a room on the United States Coast Guard vessel, the Healy, somewhere off the coast of Alaska. We'll get to them in a few moments. If you are joining us virtually, we're going to share a few things about the platform that we're using. It's called Adobe Connect. And for those of you that are joining us virtually, you will see that there is a public chat area and there's people already saying where they're from and how many people or students are with them. And we'll be sure to share that with our presenters today. The team is on a ship and they do not have the internet, so they can't see your questions or your post. It'll be my job, as well as Judy, who is also with Arcus and online today, to share questions and share the information and things that you post in the chat box to them as we go along. So many of you are already started introducing yourself in the chat box. We're having you type your name and where you're from, and if there's other students or adults that are participating with you in the same location. Again, we will share that with the team as this event is being archived, and so that they can see where you are all coming from and who's joined them today. Before I turn it over to the team on the Healy there, I wanted to explain a little bit about what is PolarTREC and why Sandy is on the Healy and why we're doing this event today. PolarTREC stands for Teachers and Researchers Exploring and Collaborating. And we've been running this program for over 10 years. It's administered by a nonprofit, the Arctic Research Consortium of the United States, a mouthful, I know. And we're a nonprofit that's based in Fairbanks, Alaska, as well as an office in Washington, D.C. We place teachers with polar researchers, both in the Arctic and the Antarctic. And to date, we've had over 150 teachers that have had experiences like Sandy that she'll share with you and working with researchers. And of course, a lot more information about PolarTREC and being able to follow all the expeditions, including Sandy's, is on our website at polartrec.com. Today, if you have questions, you can, during the presentation, type your question in the chat box and we will interrupt the team appropriately and relay the question. At the end, we'll ask you to let us know that you want to ask. Yes, can you hear me?
Yes, I can now.
Okay. Yes, please let me know if there's any other people that cannot hear me as well, or have any trouble hearing Judy or Sandy. So during the presentation, we want you to type your question, and at the end, we hope we'll able to ask questions live. So with that, I'm going to turn it over to the Sandy and the research team. And Sandy, just to give you a heads up, we have over 12 people that have joined. Everybody's introducing themselves. It looks like a variety of teachers, and it looks like a lot of NOAA Teachers at Sea alumni have joined you. I'm on your first slide, title slide, so just cue us as to when you want us to switch.
Okay, thank you, Janet. It's great to be able to share this experience as a PolarTREC teacher and NOAA teacher at sea. We passed the Aleutian Islands last night, and we're on our way. We'll be near Kodiak Island soon, and then we will be in Seward on Wednesday. So I'm going to start with an overview of the expedition and then introduce some of the scientists and then give some highlights. And then, as you said, we'll be open to some questions afterward. And next slide.
Okay, go ahead.
The Healy left Seward on the evening of July 2nd, and we took a short side trip to Holgate Glacier, so that was a great start to this amazing trip. During the transit out, we hit some multi-year ice that we weren't expecting, so that was a lot heavier and thicker, bigger, slowed things down quite a bit, and it took a few days to get through that. So we had to do a lot of adapting with the plans. After over a dozen science stations and a mooring station, we began the transit back to Seward on August 3rd. And as I said, we're scheduled to dock in Seward on the 10th. Probably one of the biggest lessons learned is flexibility, because sometimes the ice is in the way, sometimes there's fog, sometimes the equipment doesn't work as expected, but you just keep going, and that's what we've done. Next slide.
Okay.
So here's a map of our journey, and we crossed the Arctic Circle on July 7th and spent most of the expedition above the Arctic Circle. The Arctic Circle begins at 66 degrees, 33 minutes, and 46.1 seconds north of the equator. And then we crossed back over the Arctic Circle again just a couple of days ago, and now we're below north 66 degrees latitude. Next slide.
Okay.
One of the good things about this expedition is that it brought together a lot of different components to give a better picture of biodiversity and conditions in the Chukchi Borderlands area. The research areas were loosely divided into benthic, pelagic, and microbial through ice and hydrography. There was also a representative from the U.S. Fish and Wildlife Service recording data on bird sightings, and there was a marine mammal team that was collecting data on marine mammal sightings. And early in the expedition, one of the scientists, Dr. Lindsay, made a comment, and he said, "So little is known, that's why we are here." And I've kind of kept that in the back of my mind a lot as we've continued through the expedition. Next slide.
Okay.
I thought of this group quite often as our floating UN, because we represented so many different countries, as well as the United States, as well as different states and even different regions. We had Norway, Japan, Taiwan, Australia, and the United States represented, and as I said, lots of different states. And one of the highlights of the trip was seeing the different perspectives that each of these people brought. And also a highlight was seeing that using this multidisciplinary, national approach to measuring biodiversity in Arctic conditions was a good way to approach a vast area about which little is known. And we were led by Dr. Russ Hopcroft and Dr. Katrin Eichen. Next slide.
Okay.
Dr. Hopcroft is a zooplankton ecologist from the University of Alaska in Fairbanks, and his team used a lot of different types of nets, ROV, the CTD, and other tools to collect data. And Dr. Hopcroft's technician, Caitlin, was really good. Anytime something really cool came across, we were all at her microscope peering through, things like the copepod that you can see on the slide. And it was just always exciting to see what the new thing of the day was, what was going to come up, what were we going to learn or discover that day. Next slide.
Okay.
Dr. Eichen served as co-chief scientist. She is a benthic ecologist from the University of Alaska Fairbanks, and she's conducted research in both the Arctic and Antarctic. And some of the tools used by Dr. Eichen's team during the expedition were the trawl net, the box corer, the ROV, and even water from the CTD to help collect data or provide data. And the polyoid that you see in the picture on this slide was one of Dr. Eichen's favorites. And as I was looking at that picture, it just amazed me at how delicate those little plates are. as well as the color. And I think it kind of looks primeval when you look at those different plates. And it was always interesting to see the variety in life from different locations as well as different depths, because we did sample at a wide range of shallow and deep depths. Next slide.
Okay.
Dr. Lindsay is from Japan via Australia, and he is a midwater ecologist with the Japanese Agency for Marine Earth Science and Technology, and also says he enthusiastically enjoys studying jellyfish. And that's when the ROV was a great tool because it gave lots of opportunities to both view the jellyfish as well as collect specimens. And he shared a lot of his photographs, including this one of the pipe jelly. So it was always exciting to see things from his perspective as well. Next slide.
Okay.
Dr. Collins is based at the University of Alaska in Fairbanks, and he studies ice and microbes and introduced kind of a new term that he likes to use, and that's the cryome, to describe his slice of the science world. And Dr. Collins said that his interest in extremophiles led to his interest in studying organisms that make their homes in ice. And I found it really interesting that those ice channels could be 5 to 6 times saltier than the surrounding seawater, yet filled with an array of microscopic life as well. Next slide.
Okay.
Dr. Welker and Dr. Klein are both from the University of Alaska in Anchorage, and they are using water isotopes to understand changes in hydroclimate. So they use lots of different ways of collection, including the CTD and other tools, and hopefully they can use this data to get a better picture of how isotopes of water cycle through our environment to see what has happened in the past and see what happens now and perhaps what happens in the future as well. Next slide.
Okay.
Dr. Stafford is principal oceanographer at the University of Washington, and she specializes in underwater bioacoustics. She's been collecting information about marine mammal sightings while on the expedition, and she was usually able to tell what kind of seal had popped up before I had even located it in the water. They did most of their work when we were transiting at the beginning and end of the journey or between stations. And I loved this picture of the walrus. It visited the ship one day, so we were surprised to look out on the fantail and see that walrus there. Next slide.
Okay.
Liz Lebonsky is a wildlife biologist with the U.S. Fish and Wildlife Service, and she's been recording observations of birds during transits. And usually, as with Dr. Stafford, Liz had seen, identified, and recorded most of the birds before I had even focused my binoculars on them. So I get to listen to lots of different things, hear about many of the birds that make the Arctic their home as well. Next slide.
Okay.
We used a lot of different sampling equipment to help gather data while we were on the expedition. The box core collected sediment samples that were later sieved to separate organisms from the mud. Multiple types of nets collected organisms ranging from plankton to sea stars to fish. The CTD was deployed at most science stations and was used to collect data on water temperature, salinity, and depth. And we could also collect water samples from different depths as well. Augers were used to collect ice cores, and those were sliced up and or melted to give some picture of what the microbial life was in the ice. And then the ROV allowed the Pelagic and Benthic teams to view life in its natural habitat. The ROV also allowed the scientists to collect specimens and bring them back to the boat. And it might seem old-fashioned when you have kind of a state-of-the-art ROV, but you're also going to rely on things like visuals, maps, binoculars to make bird sightings and marine mammal sightings as well. Next slide.
Okay.
So pelagic organisms are going to extend above the seafloor and throughout the water column. And the group learned a lot about behavior, for example, ctenophore distribution and behavior, in large part because they were able to see these animals through the eyes of the ROV. It's really difficult to collect whole specimens of things like jellies because they get destroyed in the nets, and you don't know if you have one or lots or pieces or whole parts or whatever goes there. And this Dumbo octopus, also known as an umbrella octopus, was seen with the ROV and put on quite a spectacle for all of us who were watching. Next slide.
Okay.
Benthic organisms are going to live on or near the seafloor, and in general, when specimens are collected using nets or other tools, they all get lumped together, so you don't get a good understanding of behavior. There are few clues about that, but with the ROV, a big surprise was seeing something like this benthic jellyfish hanging out on the bottom of the seafloor. And another surprise was found in these bowl-like depressions on the seafloor called pockmarks. The scientists had wondered whether organisms in the pockmarks were different from surrounding areas, and through ROV evidence and box core and other kinds of evidence, they were able to see that life is different in those pockmarks. And this pycnogonid, or sea spider, was huge compared to sea spiders that are observed in non-polar areas. And I held this one. There's a picture on the journal, and the legs of that sea spider pretty much extend throughout my palm. So it was a pretty cool thing to get to hold. Next slide.
Okay.
Some of the challenges included unpredictable conditions of ice. As I said, we hit that unexpected multi-year ice on the transit out. Weather, fog, equipment. Even though you know you're going to the Arctic, where conditions are apt to change quickly, you still approach the expedition with an outline, and you have somewhat of a plan that you want to accomplish. Between stations or even on stations, a lot of times the schedule was constantly updated and adjusted so that maximum range of equipment could be deployed at each of the stations. We were in a remote location where there weren't any places to get spare equipment, so pre-planning, flexibility, creativity, all of those things that we emphasize in the classroom were especially important. Next slide.
Okay.
As I've been reflecting kind of through the whole expedition and over the last few days, I get asked a lot, what were the highlights? What was the most important thing or the biggest thing? And it's really a tough question because there's not a single favorite animal or a single favorite moment. But I think for me, a highlight of the expedition was seeing so much diversity of life from microscopic to polar bears. Sometimes you look out at that ice and it seems like a barren wasteland. You just don't see anything that's big and obvious, but then you bring up a container of water or a net's contents or a bucket of mud or even a chunk of ice, and you see this amazing array of life. And it was pretty cool to notice how things changed with geography. So when we were deep in the midst of the ice sheet, there didn't seem to be a lot of life above water, but as we began to move southward during the transit home, once again, the life above water increased. The ROV was amazing. We got to see and learn about organisms in their natural habitat. And I was also really impressed with the way the group worked together in spite of challenges. As Dr. Eichen was saying, 6 weeks can be a really long time if you're stuck on a boat with a bunch of grumpy people. And instead, the trip went by very quickly. So there's some laughter in here. But the people on board made the trip go by rapidly. I was so privileged to have been a part of this expedition. And from learning about and getting to use different kinds of equipment to meeting people with all kinds of backgrounds to seeing how all of the pieces of the expedition fit together, it's been something that I can't even put into just a small sentence. It's so much bigger than that. And I think the message of the Arctic is so much bigger than that as well. Next slide.
Okay.
That brings us to questions. Would you like me to introduce the scientist, or how would you like to do that?
This is Janet. So, yes, please introduce the scientist. And yes, this is Janet. Can you hear me, Sandy? Sandy, can you hear me? Okay. So, please introduce the scientist. And while they're doing their introductions, I would really like to kind of backtrack a little bit. And for our audience, and have maybe each of the scientists talk a little bit more about why they are on the Healy and why their interest is in the Chukchi Borderlands. What was this expedition all about? Looks like it was cool. There's lots of people that are collecting science, but who cares? Why should we care about the Chukchi Sea Borderlands? So as you go around the room there and introduce scientists, please have them tell us why this is important research and why they're there. Thanks.
Okay, we will start with Dr. Hopcroft.
Hi, I'm Russ Hopcroft, and I work on Zooplankton. And we are here because this is an area of the world that is very poorly known, difficult to get to, and it is undergoing pronounced change as sea ice melts and the climate warms, and we think it's valuable for us to understand this system before it changes more than what it might have already.
And this is Dr. Eichen.
Yeah, this is Katrina Eichen, and as Sandy explained, I am a benthic or seafloor ecologist, and in addition to what Russ just said about being an unexplored and very unknown region, there's a lot of topography in this region, which is particularly interesting for seafloor organisms. So we have valleys and canyons and ridges and those kind of things, and then these pockmarks that Sandy mentioned, which are sort of bowl-shaped depressions in the seafloor made by extinct gas seepages. And so there's a lot going on the seafloor here that we oftentimes don't see in other regions. And so from that perspective, it was really interesting for the seafloor ecology.
And we have Dr. Lindsay.
G'day and konnichiwa. Dougal Lindsay here from Japan via Australia. I work on deep sea midwater plankton, mostly on gelatinous animals that are normally destroyed when you try to capture them by traditional means such as nets. I'm here with this expedition in the Chukchi because this area, as Russ and Katrina have both said, is changing rapidly, and we need to get baseline information on what the system is like now before things change too much, but also because the input of food into the system is mostly only during several months when it's warmer, and the ecosystem under the ice could be very different from that in other areas because of this. And we had assumed maybe there were many gelatinous animals which don't need a lot of food to survive, being mostly made of water. And we've seen that this is indeed the case, and there are many new, exciting new species and discoveries we've made this trip. Thank you.
Thank you. And we have Dr. Klein.
Hi, Eric Klein. I'm at the University of Alaska, Anchorage, and I'm a geoscientist who specializes in the movement of water, trying to understand how the water cycle behaved in the past, how it's behaving now. And I use isotopes as a tracer, or basically a measure, to help us understand how water is moving. And to echo some of what we've already heard, I'm up here because it is an area that we don't know very much about. And if we have more samples and understanding of these signatures of how water is moving through the Arctic, we can understand how it's going to change. This area is changing rapidly, particularly as the sea ice cover. And with changes in sea ice, you're exposing a lot more water to incorporate into the environment, and we're looking to see how that is going to unfold in the future with these changes in sea ice.
We have Dr. Welker.
Hi, it's Jeff Welker from University of Alaska, Anchorage, and Eric and I are working together on aspects of the water cycle as part of this mission, and using a couple of new isotope techniques to measure these at very high frequencies over large distances. And so one of our interests has been, how does the isotopic characteristics of seawater and the vapor associated with the marine boundary layer, how does that vary over large distances between the Gulf of Alaska and into the Arctic Basin? The other aspect that we're quite interested in is working with Russ and Katrin on understanding the water aspects of the food web. And so historically we've worked and focused in large part as a community on the carbon and water, carbon and nitrogen aspects of the food web, but we're attempting to do more in terms of understanding how the water cycle may imprint itself regionally on these different food webs.
We have Dr. Stafford.
Hi, I'm Kate Stafford from the University of Washington, and like everyone else, I'm leading a marine mammal survey to look at changes in a region that is very remote that we seldom get to go to. And marine mammals are a really good ecosystem sentinel for changes in the ecosystem. They feed on benthic animals, they feed on zooplankton, they feed on fish. So all of the changes that everyone has just been talking about may be reasons that we are starting to see a different species composition in the high Arctic. And so we wanted to get to this area that no one gets to go to document what animals might be there now for comparison with the future.
We have Liz Lebunsky.
Hi, my name is Liz Lebunsky. I'm a biologist with the U.S. Fish and Wildlife Service based in Anchorage, Alaska. And I'll just follow up on what Kate said. Much like the marine mammals respond to the environmental changes, the seabirds do the same thing. So we're really excited about having this opportunity to go work up in the Borderlands region. It's quite a remote area that we seldom have had an opportunity to survey. So I was very excited to conduct seabird surveys now, and with the potential idea as change does occur in the high Arctic, how seabirds may change their distribution and feeding patterns in the future.
And that's our group of scientists here today.
Awesome. Very nice. Can you hear me now again, Sandy?
Yes, I can.
Okay. So we did have some questions that came up, and I'll relay those in a moment. Just one question I have is, do this group of scientists do this every year? If not, how often do you go out on the Healy or plan on, I don't know if you're planning on continuing this type of survey as a group together or not. So maybe somebody can ask that while I get questions.
Well, the last expedition we had like this was 11 years ago. So a big operation like this is expensive, and therefore it's not the kind of thing that happens very frequently. The Healy itself only has about 3 months a year, 4 months a year that it operates in the Arctic. And there's huge competition for scientists to get time on the Healy to do the various types of science that one could do in the Arctic.
Okay, great, thank you. So there was a question that came up. It was, I think, when Sandy was going through stuff. Let's see if I can remember. Oh, Julia West would like to know, can you explain the ice channels that are so much saltier? What is that? Sorry if she missed it earlier.
Yeah, this is Katrin Eichen, so I think I can take a stab at this. So sea ice obviously forms when the seawater freezes, but what really freezes is the water part, and it expels the salt part of the seawater during the freezing process. And it creates a very salty brine. And during the freezing process, this brine is then concentrated in little challenges within that ice matrix. And oftentimes it gets trapped there. And so that is why the brine channels within the sea ice is so salty. And as Sandy said, I mean, it is incredible. And for the most part, you would think that no organism would be able to live there. But these tiny little microscopic life forms have found adaptations for being able to withstand these very, very high salinities.
All right, cool. Sue Supako would like to know, she wants to know if they're able to contact you from their classroom, meaning all the scientists, They study marine invertebrates, and each child gets to select a cool invertebrate to study, and having a scientist communicate with them would be fabulous. So they're looking to see if you would all be agreeable to being contacted by her.
I think our contact information is all on the website for this expedition.
Yes, it is, and we'll relay that again to it. So I think that would be great. Julia West has a question for Russ. She wants to know, have you seen any reductions in copepods because of ocean acidification?
At present, there's no connection between the population size of most open water zooplankton species and ocean acidification. It's a difficult question to really get a handle on because we've really only been looking at it as a measured parameter for a relatively short period of time, and therefore it's hard to have collections over a long period of time, ocean acidification measurements, to see if there's any correlation between them. There's no hit-you-in-the-face response that we would currently attribute to ocean acidification, which is also happening at the same time as a certain degree of climate change and global warming. So the two of them will be incredibly hard to tease out as we move into the future.
Okay, great. I was going to tell our audience that if you want to ask your question live, just click on the little hand or person up at the top, and then we can, or just let me know in the chat that you want to ask a question live. In the meantime, I was going to ask Sandy, can you give us a little bit about what it's been like to live in Lake Healy and how many people are there? And somebody was asking about ice. Hold on. Oh, did the seas get rough? So can you tell us a little bit about life on the Healy and working out there?
Okay. Life on the Healy is actually pretty cushy for a research environment. There are usually 3 of us in a room. The Coast Guard supports everything that we're doing. So we have a galley where we go for meals. It hasn't been terribly rough until the last couple of days because we've had so much ice cover. But when we came out of the ice, then it became a little bit rougher. It's cold when you're sieving mud out there or bringing in nets or doing things like that. I think on the Healy there was a good comment. It said that the inside temperature responds to the thermostat, but the outside does not. So inside the Healy is pretty comfy. Outside can be pretty cold, but today it's 58 as we're headed to Kodiak. So the conditions change daily, or momentarily, I guess is a better term. Did that answer what the person was asking?
Yeah, I guess they wanted to know, so since you're out of ice right now, it's not too rough, and since you're out of ice right now, it's not too rough, and did anyone get seasick or things like that?
There are 140 people on board. It's actually rougher now because we are out of ice. There was some seasickness going out. There was some seasickness coming back. I have not experienced it, so I'm fortunate. But it's been there. But generally, it hasn't anyone from doing what they needed to do for a very long period of time. Fortunately, there are patches and medications and things like that, and we have some medical personnel on board as well.
Very cool.
And I can maybe just one thing from the science perspective and living, on a research vessel like this. that might be of interest is that we work 24 hours a day, and so it's basically around the clock activities, and so one of the things to get adapted to is not having necessarily a regular sleep schedule. You get up whenever you need to get up, and if it's 2 o'clock in the morning, so be it. And then sometimes it's not an 8-to-5 job here, but sometimes it's very long hours. And I was always amazed when I was in the lab and it's 3 in the morning, and all of a sudden shows up and says, "Well, the box score is coming up. I'm here to sieve." And it's like, wow, yeah. But that's just part of the life here, where we also need to make the most of the time we have here.
Very nice. You guys will. Go ahead.
The 24 hours of daylight was harder to get used to.
That's because you don't live in Alaska. We like it. I have a couple of more questions for you from Sandra Brown, and she's a language arts teacher. She has a question. She wants to know, what are some meaningful ways a language arts teacher could incorporate information about the Arctic or this experience in general? Are there resources that you could point us out to make this engaging for kids? I will put the PolarTREC website, but if you have some thoughts about that, Sandy, that would be great.
Obviously, as you said, first up, PolarTREC website. NOAA Teacher at Sea actually has a lot of resources as well, but I think it's a way to get students excited. So it's a way to use language arts in a way that gets students enthusiastic and excited about the world around them. So I think cross-curriculars, working with a science and language arts team. I'm a huge proponent for as much writing, real-world writing as can be incorporated. And Dr. Lindsay has something I think he wants to add to that.
If arts there would include drawing and painting or these kinds of things as well, we've been taking many photos and videos of some of these strange animals that live beneath the Arctic ice. And just by looking through some of these images on the website, the students should be able to be stimulated by the range of different colors and forms and habitats and ways of living there are there, and could maybe incorporate that into either their pictures or into their writing as well.
I would agree with that. That's great.
Yeah, I would agree with that too. So here's another question related to that. Sue would like to know, how will you use this, Sandy, in your classroom? Are you going to have any lessons for elementary school?
Yes. Before I answer that, Dr. Welker wanted to add something about the last question with the language arts connection.
Jeff, I was just going to say something about the language aspect that could be really valuable would be to maybe introduce the students to some of the Inupiat words for the different environmental conditions that we see in the Arctic. So they, not that I know them myself, but I think it would be an interesting exercise to see if they have different terms for different sea ice conditions, for instance. We're having an assistant. One of the other colleagues here has got a suggestion she wants to say as well, so let Kate. But I think, again, the language in describing the Arctic that is native to Alaska could be a very interesting way for them to learn and think about the Arctic as well.
Sure. This is Kate Stafford. I was just writing Jeff a note pointing out that the word Sikuliak, which is the name of the new research vessel that is at UAF, it means new ice in Inupiaq.
Yeah. Nice.
Okay. Thank you. As far as incorporating into my classroom, I'm working on the PolarTREC lessons that will get posted through PolarTREC website. And I teach at a pre-K-12 school, and I think teachers can adapt lessons. So generally what I do when I provide lessons in our school is give suggestions on how to make them for lower school, middle school, or upper school. So for example, with albedo, looking at melting ice and it's just a feedback setting that causes more ice to melt, I've done those activities with first and second graders using some simple light probes. And first and second graders can collect data, and they can begin to look for patterns in that data. Some of the other plans that I'm looking at, graphing, the first and second graders might not be able to produce complex graphs, but they can start to look for patterns in those graphs. I'm doing some things that simulate a box core, and I'm trying to put that together. And I'll be glad to share anything that I've developed for my classroom with the questioner if they just send their contact information. And they can send that through the PolarTREC website if they want to.
Excellent. Thank you. I also said that if they had more questions about resources and if some of the researchers have specific links or resources that they want to share, have them give them to you, and then, you know, we can post them. You can post them through a journal or just email everybody about it. So, because there's lots of great ideas. I do like Jeff's idea about, you know, incorporating that indigenous perspective because there's a lot of information out there. And they said, Sandy said to say thank you. We do have a question from Rachel Large about birds. How were you able to identify all the various species of birds? Do you use visuals or sounds or combinations? Never mind. I hear the heel. Okay.
We're in the middle of some drills. Sorry, to repeat the question.
I will. I figured it was somebody from the Healy talking there. How were you able to identify all the various species of birds? Do you use visuals or sounds or a combination?
Well, in this case, while we're doing observations on the Healy, we're actually indoors conducting our surveys from the bridge. So we are limited to the visual cues for identifying the seabirds. One of the major things that I look for when I first spot a seabird is the bird's actual movement or behavior, trying to see what its flight pattern might be, and also the major coloration patterns. That usually helps me key in what type of seabird I'm dealing with, whether it's an alcid or a shorebird or anything along those lines.
All right, excellent. That kind of related to another question. I forgot who said they were looking at different species through the ROV. How many new species were discovered?
I'll take that.
Oops, we lost you. Uh-oh. Do we have the Healy? The Healy? All right, I think we just lost the Healy. I was surprised that they stayed connected as long as they did. We'll let them rejoin here, and we'll hear from Dr. Lindsay about the number of species. But if you have questions that didn't get answered or things that you're curious about, go ahead and keep typing them in the chat box, and we'll be sure to relay them here in just a moment. In the meantime, if you joined us a little bit late, this event is being recorded and we will be archiving it. And the archive will be posted on the PolarTREC website, and we'll also share it through through everyone that signed up. Is that the okay, hi. All right, we were asking. Yes, I hear you.
Okay, for species, here we go.
Okay.
This is Dougal Lindsay from Japan, Australia. At least for Japan, Australia. This city on the Northern Island. I've got Sydney in Australia, and you've got it in Canada as well. So we found probably around 6 new species of gelatinous animals while we've been up here. Some have been even more exciting than just species, but even higher taxonomy levels that seem to be new to science. So we're looking forward to going back and checking and rechecking that this is indeed true, so we can introduce them to the scientific world in general and to everybody else as well.
Okay, great. Thank you. Let's see. There was a question from Cara, and she wants to know, Sandy, what was your favorite new piece of equipment to use while working with a different scientist?
I really liked working with the CTD operator because when you're looking at that, as the CTD is descending, you can see certain markers, and that helps to determine where you want to test samples. So I thought the way that all of that worked together was pretty cool. Of course, the ROV was amazing because you could see things. You could just look at the water column or the seafloor. But then I got really good at sieving mud. So I don't know that I could pick a single favorite because there were so many different things. But the one thing that I did like really the best was the way that the lab equipment, the lab techniques that I teach in my classroom are applied across the board. So I saw them being used on a much different scale, but same kinds of classroom techniques.
Okay, very nice. Good. All right, you're getting some thanks. And I'm looking to see if anybody else has any additional questions for you. So just hold on a second. While I'm waiting to see about questions, when did you say you're getting back to Seward?
We are scheduled to dock in Seward Wednesday morning.
That'll be exciting.
Helicopter off tomorrow.
Very nice.
This is Judy. So
ahead.
because these are so rare, does Russ or Katrine have any idea when another research trip will go on like this again?
Well, this is Katrine Eichen. So all of us are doing field research nearly every year, but not necessarily in this group and not necessarily in this region. Most of us work a lot in the Arctic, but a lot of us also work in other regions, and so it really changes, and what we do every year. But this was a pretty unique opportunity for all of us, as we started out saying, in terms of the combination of disciplines and people as well as the region.
And then there was another question. Are you there? Okay.
Yes, we one more comment on that.
One more comment here. It's Dougal again. I'd like to go especially and look at the Atlantic sector of the Arctic, where it's also rapidly changing, but changing in a different way, because there's a much deeper channel between the Arctic and the rest of the world's oceans, and compare the two to see exactly how different they are and what is related as well. So although we may not be getting together on the Healy again in the very near future, there may be other expeditions that will allow at least some of us to go off and use the data we've gathered here to learn more about the Arctic, maybe in different sectors as well.
a couple of things that came through while you were talking. There was somebody that wants to know, Emily wants to know about the polar bear sightings that you saw, Sandy.
Okay. I'm going to let our mammal person speak to that.
Okay.
Hi, this is Kate Stafford, and we had 9 sightings of a total of 12 polar bears. So one of those sightings was a group of 3, which was the mom with 2 2-year-old cubs. And all of the bears we saw looked really healthy. We were taking some data on body condition for some people at U.S. Fish and Wildlife. And we saw them in the ice, generally in thicker ice. But Liz saw our last 2 sightings of polar bears when she was up on the bridge at 3 in the morning getting the last view of ice. And the very last bear was sniffing around a few walrus on an ice floe nearby.
Wow.
And I would say that maybe our best polar bear sighting that the whole cruise got to see was when we were stuck in ice. A very curious young bear came bounding over to the Healy and put on quite a show for everyone.
That's exciting, very exciting. So Sandy, Sandra Brown wants to know, how does this compare to India?
Oh, Sandra. Okay. Well, it's slightly different. The common link is water. I think the excitement of learning and thinking about how to put it into a classroom ties the two together. And when I was in India last year, the common theme was water. And in the Arctic, what's happening with the water is a common theme. So tying together how climate changes affect the world and how we are much more connected than separated has been a common theme.
That's great. Very good. You got some kudos for that one, Sandy. Nice response. Okay, let's see. I'm looking through, make sure we got everything. Oh, I had a question for kind of the research team. Like, how long does it take you to analyze your data, and when do you start kind of sharing that with everybody? What's kind of your plan for sharing data?
Well, we are already analyzing data. One of the things that happens at the end of the cruise is just the basic record keeping, getting your books in order, making sure all the information you have is complete, tidying up data records, and some of the analysis can be done based on observations that are here, and there's a lot of analysis that involves going through samples that are already curated and will be reopened once we get back to the labs. I would expect that part of the operation will take on the order of another year before we can start writing up the papers. Some things will happen much quicker.
All right, excellent. Okay, I see someone typing here, so I'll wait for that. And then otherwise, we're coming up on our hour, and I want to thank you guys, especially the team there and Sandy for dialing back in and doing a great presentation. And we are archiving this, so we'll be sure to share that with everybody. We just got a comment that says, wow, I didn't realize the research would be so long ongoing, a year. I'm impressed. So yeah, it was good to hear how long it takes for you to kind of wrap that all up. So any last or parting thoughts from Sandy or the research team?
What an amazing area, and the Arctic seems so far away and remote from coastal Virginia, and it's easy to think that, you know, what you do in one area does not affect another area, but it does, and those connections are pretty strong and pretty amazing.
All right, thank you. Anybody else?
I think we're good here.
All right.
Just because Sandy and the crew can't see it, there's all kinds of great comments coming in. Michelle Eno, this has been awesome. Thank you all. Sandra Brown, fantastic information. Almost like everyone should get to experience this. Turtle Hayes, thank you for the things to think about. And Sandra Brown, quite an eye-opener. And Kara, the Chick is thanks, Sandy, and everyone on the Healy. And Maggie Kane, great job, Sandy.
Thank you.
Oh, one more.
Right.
Emily Cisco says, have a safe trip home.
Thank you. We will. Intent.
All right, thank you from the Healy, and we look forward to hearing more and reading your last little bits of journals as you get home.
All right. Thank you.
All right. And for everybody else,
Everyone.